Alrighty, folks, today we are talking about cardiac contraction, which is basically electrical impulses which make the heart go. We're just gonna talk about that real quick. So, just as a brief overview or review, I don't remember, I don't know, thanks. Let's talk about the contractions of the cavities of the heart themselves. So, if we have these little candies, and these represent blood cells, then we have blood enter in the right atrium and the left atrium. There's a contraction which pushes these blood cells through the valves, contractions in the ventricles, which then push the blood cells through the pulmonary and systemic circuits throughout the body. So we have blood come into the atria contract, push blood into the ventricles, which contract and push the blood throughout the rest of the body. How we get those contractions, that pushing motion from the atria and the ventricles, has to do with cardiac contraction. So, we have an electrical impulse. When we talked about our skeletal muscles, that electrical impulse came from the brain. And so whenever we do any contraction motion with our muscles, that is electrical stimuli that produces or promotes the movement of calcium into the motion, right? So we have our actin and our myosin filaments, and calcium comes in due to that electrical impulse and forces our muscles to contract through that actin and myosin sliding filament theory, right? So we talked about that. Very similar with cardiac contraction, with the calcium promoting that sliding filament theory between the actin and the myosin, and pulling those cells and those muscles together. However, the heart is self-sufficient. The heart will produce its own electrical impulses, and it does that through these things called pacemaker cells. Pacemaker cells. And these are myocytes that produce action potentials. So, electricity. Those are gonna there are two spots. We have one here, and we have one here. One in the right atrium and one on the right atrium, right ventricle border. We're really only gonna talk about this one and just in the extent of this spot. This is called the SA. This is a bundle of pacemaker cells that push those action potentials through the heart. We have electricity. Spike in the heart monitor. So right here. And then we can say that the electricity kind of diffuses, right? And so remember that energy is never lost or gained. We can't make it and we can't lose it. We can't destroy energy. So what happens is this electrical energy, this electricity is converted into kinetic and chemical energy, and so that's the contraction. So this electrical energy turns into kinetic and chemical energy, which contracts the heart, and this energy is diffused, and so the ventricles go back into their relaxed state, and so we see this kind of bill again on the EKG or ECG. So whenever, like, if you watch movies or if you go into the medical field and you're familiar with this, this is that bitty sort of dealio going on with heart rhythms. We're measuring the electrical impulses and the contraction of your atria and your ventricles. At a cellular level, it's absolute nonsense. Like, you have all of these cells hanging out, joined together, and it's a mess. So what we're going to do is we're going to break that down step by step. So we're going to call these SA nodes, these myocytes, we're going to model those as little boxes with a little circle inside. And this little circle gets to hold all of the ions, which I am going to mark in red. So we have red dots. These pacemaker cells with actin and myosin bundles. And so if we model several of them in a row, if this is a pacemaker cell, it does not contract, and it is autorhythmic, which means it produces its own rhythm, it produces its own electrical impulses. It doesn't take anything from the heart or from the brain. It's self-sufficient in the heart. It doesn't take anything from the brain or the nervous system. These are myocytes that do contract, and these are non-auto. Oh, I spelled that wrong. Rhythmic. These are non-autorhythmic because they take their energy from the pacemaker cells. So this autorhythmic cell shoots energy through the myocytes that do contract, which are also non-autorhythmic because they take this energy from the autorhythmic. And so these are joined together by what are known as gap junctions. So we have our ions in here, they move in here, and these are, we're going to say these are calcium ions, right? Because we talk about calcium promoting that actin and myosin filament, sliding filament theory. And so it'll travel through this myocyte, travel through this myocyte, travel through this myocyte, through these things called gap junctions. Because this is a gap and it joins it together. It's junctioned, it's a gap that's junctioned, it's joined gap. Easy peasy. Easy peasy. And so, right, we have our pacemaker cells all along this area. So in the blue, in the green, in the blue, and the orange, we have our pacemaker cells along here, kind of. And then we have all of our myocytes that go in the cell. So we have all of these guys kind of on the outside, if you want to think about it that way, along this path of the action potentials that then trigger the ionic charge through the myocytes that do contract, the rest of the heart muscle. let's talk about what that electricity looks like and why. So I asked you to watch that video over electric eels and electric fish because I thought that video did a really good job of explaining the electricity that passes through those fish bodies, which is very similar to how that electricity is created in our hearts. So, as a quick reminder, you have your fish. I'm going to draw my fish in green. You have your electric eel fish. Hello, he's happy today. And he has down here a lot of different cells that if you take a bigger look at are polarized. So you have positively charged ions here and negatively charged ions here. And so what happens is if it undergoes sudden shift in polarization, then it sends out that electrical charge to zap any prey that it's looking for. And very similar with like a balloon. If I take this balloon and I rub it on my hair, it is now more negatively charged. So it now has an electrical current running on the outside of it. Not through it because it's rubber, but on the outside of it. So now it's negatively electrical charged, and it'll make this balloon do a little bit of a motion. I don't even need to touch it because of the electrical field that's going on. So if I make this as still as possible, and then I bring this guy in closer, you can see that this balloon starts to move around due to those differences. So you have one balloon that is now negatively charged, and another balloon that is both positively and negative charged. And so this balloon will start spinning around to get the positive closer and the negatives further away. Remember, opposites attract. And so that's kind of what's happening. And if you like rub your socks on a carpet and then go zap someone, that's just sudden shift in all of these. So in your cell, in all of your cells, you have your cell membrane, and you have tunnels for ions to pass through. Ion channels. So this is like an ion channel, and let's say it's for sodium, and this one is for calcium, and this one is for potassium. What you have is, I'm going to say, all of these pumpkin seeds are going to be sodium. All of my black-eyed peas are going to be calcium. And all of my regular pinto beans, woo, there they go, are going to be my potassium. And so what's happening is that these ion channels will pump, the calcium ones will be pumped out, the sodium ones will be pumped in, and then all the potassium ones will also be pumped inward, I believe, if I'm telling you correctly, which God, I hope I am. And so you have this change in ionic energy, right? Because you have the positive and the positive, and then you have this 2 positive. So these positive and positives are relatively negative. So we're going to pretend these are negative ions, and this is a positive ion. So what's happening is your cells are pumping all of the positive ions onto one end, of your cell. So on the outside, let's say, and then all of the sodium and potassium ions inside the cell, and it's pumping these guys in and pumping these guys out. So in, and then what happens is there will be this shift of ionization. So all of these either switch places or like mix around, and that sudden surge of change in polarization is what causes these contractions. So you have positive and you have negative, and then you suddenly have them depolarizing. So you have positive and you have negative, and then you have them mixing together, and that is that contraction, that electrical impulse that we see in the heartbeats. So the electrical impulse, it starts up here with all of the calcium ions on one end and all the potassium and sodium on the other end, and then they mix up here, and there's that contraction, and then it goes down here and they all mix, and there's that other contraction, and then they all go, and they're all separated again, and that is that repolarization or relaxation of the ventricles. We don't see the repolarization of the atria because it's so small and hidden by this much larger depolarization or contraction of the ventricles. So, this is a confusing topic, just letting you know. So if you didn't understand, that is okay. Just let me know what questions you have. We will continue to study this throughout the week, and hopefully you'll get a much better understanding of cardiac contraction after this. All the best.